Yeah, thanks. So I think we have a ton that we can talk about today. I think very generally, our topic is on genetic engineering, genome manipulation technologies, sequencing, and where everything is going in the future. So feel free to add questions in the Q&A or the chat, and I think we'll try to address them towards the end. But if they come up, if things are relevant in the meantime, I'll try to keep an eye out for them. So I thought maybe we could start, since it's so broad, with a little bit more of a specific topic and then go out from there. So maybe Vivek, you can talk first. And I was wondering if you might explain to the audience basically what you're doing with bacteriophages and what are you trying to characterize? Why is it useful to us to know a lot about bacteriophages and what can they potentially do for us?
Thank you. One of the maybe I'll start with the really basic. So bacteriophages are viruses which kill bacteria. So these are healthy viruses, or helpful viruses rather, unlike SARS-CoV-2. So these are viruses which help in maintaining the microbiome dynamics that we see, or evolution or adaptation to different stress conditions that we can see microbes are adapted to. And one of the challenge has been, even though we know bacteriophages are everywhere, from ocean water to human gut microbiome, we don't know exactly how do they really do their job, or what do they do actually to impart the functionality. So one of the challenges is to understand the function of these viruses, and not only that, how do we use them for applications? And one of the key focus of my work has been using them to understand how the genes are, what are the genes of these phages are made up of, what do they code for, how do we engineer them, how do we recode them for applications like eliminating a pathogen in a community of microbes. One of the challenge here is to understand, one, not only coding, what do they code for, but also understanding how they interact. What are the resistance mechanisms? If we are thinking of a phage therapy or a solution for antibiotic resistant microbes, the challenge has been we don't have any knowledge, much of knowledge, how the interactions are happening and what resistance mechanisms. So my research focus of my group has been developing tools and technologies to untangle these challenges of foundational research, where we're trying to understand the gene functions, also understanding how they interact with bacteria, and then using that knowledge to engineer those viruses. And right now, we don't have any mechanism or technologies to do this at scale. I can go into the deeper levels, but let's keep it, if that is satisfactory.
Sure, yeah, that was great. I've always been super interested in phage therapy ever since like undergrad. I would be reading about it, and I'm often a science fair judge, and a lot of times groups are presenting phage therapy as their solution. It seems like a very popular idea. So I was curious if you could talk a little bit about what phage therapy is, and why is this something that so far seems to be used in other countries and hasn't really taken off in the US, and what might we need to overcome to get to that point where it happens here?
That's right. Yeah, I mean, it has been discovered like 100 years back right now, just before the antibiotics revolution happened. So one of the recent report, I wanted to just cite that, which says that in 2050, we will have more people dying from antibiotic resistance infections than cancer, for example. And that is a huge number. And now we appreciate that number because of the situation that we are in with the COVID infections everywhere. So what happens is antibiotic resistance is obviously going to happen with evolutionary pressures. We don't have any new tools. We don't have any new antibiotics, and we don't have any other alternatives. So bacteriophages are one option, which are one opportunity which we can have to use them for infection treatment. So phage therapy is essentially using phages to treat infectious organisms, or whether it is to kill a pathogen or to manipulate a microbiome towards the helpful applications. So typical people have used phages for a long time. And one of the challenge has been understanding the resistance has been lacking. Then secondly, there are some IP issues on the industry side. There are applications-wise understanding how do we run the clinical trials for phage therapy. So these are different from antibiotics sort of, but at the same time, powerful, and still there are a lot of gaps in the application itself. So understanding the foundational knowledge is lacking, and then also the application in the clinical space.
Great, thanks. And if either of the other panelists want to chime in about antibiotic resistance or phage therapy, feel free.
Well, maybe I can ask Vivek. Vivek and I go way back. So maybe Vivek, you could tell us like what makes the field think that phage can be better at preventing or overcoming resistance than classical drugs.
Yeah, so one of the these are recently published compassionate usage examples where the hospitals are relieving patients not because they're getting better, they're relieving them because there are no medicines. So the medicine or antibiotics available for these patients. So for example, patients which are resistant to colistin, there is literally nothing the doctors can do. And in such situations, phages are given to these patients. And there are multiple examples now, published and unpublished data sets available where phages are given and people are getting better, and they go back home and they continue their life. So there are examples such as this. There are countries like Georgia Republic, they have a much more systematic over-the-counter phages available for a variety of things. So I think it has been not adopted so much in the West in terms of, especially in the US. Things are slowly changing, but I think there are opportunities to replace or at least use as an adjuvant along with the small molecules. So not only small molecules are, we don't have a new pipelines in the space. Economics are issues. Antibiotic stewardship an issue, and in that sense, it's good for one cause, but it is not helping the patients. So phages fit in that space where we can have new solutions along with the small molecules.
But do you think that if phages were to become kind of the predominant therapeutic mode, would we run into this same game where we are behind bacteria developing resistance? Or do you think there's something special about phage where it might not be as difficult, and maybe we can even evolve the phage
that's absolutely
in collaboration with bacterial developing resistance?
Yeah, I think you nailed it. I think the advantage of phages are they're limitless. Only limit is our human capacity to find them. Phages are everywhere. We can find phage for any bacteria that we can grow. We can apply engineering tools. You are good at it, evolutionary pressure, so that we can evolve these phages such that we can apply these for any bacteria which is resistant to any drug. So these have it's like a platform tool somebody is given to us so that we can eliminate these pathogens. It may not, in my personal opinion, it may not completely replace small molecules. It can be a very good adjuvant rather than on its own. But I think there are some new engineering technologies and machine learning algorithms people are trying to understand and use them so that we can prior we can understand whether there is a resistance going to happen and how do we eliminate that resistance. How do we identify a phage when the sequence is available for a pathogen? So there are a lot of new technologies and tools being developed. I think that might address these questions, and including the tools that you guys have developed in your lab. I think they are super powerful, evolving these phages for different applications.
Cool,
so Vivek, a quick question. Phage are so specific to the microbe that the specific bacterial they'll attack. So when you have an infection, how rapidly do you think you can find the right phage or isolate a phage from that pathogen to then make it a therapeutic? Yeah,
that is always the challenge, right? How equipped are we, right? So if you look at US strategic stockpile that we have been hearing in the press, they don't have phages. So let's imagine there is a population which is resistant to antibiotic, infected with antibiotic resistance. We don't really have a capacity. So coming back to your question, how do we really develop these tools? So essentially we need a national level of some kind of a phage bank or a registry where tools are developed. There is a centralized facility where people can go and take these phages or characterize them in advance so that we know what are they coding for, what do they bind, what do we know them in terms of a deeper level, like functionality of these phages, and so on and so forth. So we need a sort of a phage bank at the national level, which can be deployed on demand at a scale possible. Yep.
Great. So Chang, perhaps since Vivek kind of alluded to what you're doing in your lab, you could expand a little more on that and describe what you were evolving, why, how it works, and where you see things going.
Yeah, sure. So yeah, my lab, as I said in the introduction, focuses on developing genetic systems that can evolve very rapidly. And I think the critical challenge from kind of a platform technology perspective is that if you think about modern organisms, right, and I'm talking about, you know, bacteria and yeast and not just animals, for instance, they've all developed very large genomes. And so when an organism is asked to evolve, they have this like gigantic set of information that they must ensure that they won't destroy. So that kind of puts a cap on how fast you can explore new sequence space, right? You can imagine if I like, you know, the basis of evolution is that it's this trial and error process where you have to make errors to try to discover new things. And so if you have a very high rate of error, but you have this massive genome that you're putting that error on, you're almost certainly going to destroy something important, right? There are more ways to destroy important things than there are to find useful new biological properties. And so the kind of main contribution my lab has made is that we've made cells that contain not just their you know, standard genome, but we've created an additional genome that's replicated independently of the large cellular host genome, where that replication system is what we call orthogonal. So it doesn't interact with the genomic replication system. And as a result, if you put a gene on this specialized genetic system, it's maintained and propagated completely independently from the host genome. And because of this independence, we can dial up the error rate of this new system by orders of magnitude beyond what you could do on the genome. And as a result, you can start sampling new sequences at a much faster rate. And so what we typically do now that we have this system in hand is we put some gene of interest, usually encoding for some protein, and then we hypermutate it, or it continuously hypermutates, and then we select for new function as it's doing so. And so the types of problems that are most exciting to us currently, at least, is are in the areas of enzyme evolution or enzyme engineering and antibody engineering. So antibody engineering is something that's very relevant, especially in the COVID crisis, but even beyond that, right? Like anytime you see a drug advertised on TV that ends with, you know, MAB or AB, it's an antibody drug. And what you typically need to do is rely on like discovering antibodies, perhaps through immunizing animals against certain drug targets in order to get that therapeutic candidate. But that process is very slow, and we're basically putting antibodies onto our system and very rapidly evolving them so that they can bind and neutralize additional drug targets, such as ones associated with coronavirus. So that's kind of an antibody therapeutic application of our evolutionary systems. And then the other area I mentioned is enzyme evolution, where, you know, natural enzymes, of course, can do amazing chemical transformations, but they're typically not making things that at least the user specifies, right? I mean, certainly there are many enzymes that make things that we value, but there are a lot more chemicals that we want, and a lot more drugs that we want, and a lot more chemical transformations we want than what nature immediately provides. So we basically put enzymes onto our hypermutation evolution system and then pressure them to evolve those new chemistries. And this can be used for basically synthesis of drugs cheaply and greenly, as well as synthesis of commodity chemicals. And there are some other applications in, for instance, even like plant biosciences and agriculture that we're getting into. So, I think the exciting thing for our lab right now is that we have kind of this evolutionary engine to discover new properties, mostly in proteins, and we're applying it pretty broadly.
Very cool. I'm curious how you, or maybe all the panelists, really see the balance between kind of a preconceived, biased engineering process that is just one person in the lab thinking, oh, if I mutate this residue, maybe my enzyme will improve, versus using one of these less biased directed evolution type approaches. Do we have, winners on each side? Is it just what you're interested in? And is that why you pursue that? Or what do people think about balancing those two approaches?
Yeah, well, I'll take a stab at it first, and then I'll turn it over to others. I mean, I suppose, we're putting our money where our mouths are and focusing on the evolutionary approach. Because really, if you think about sequence space, right, like the beauty of biotech right now is that, at least in terms of the organismal side of things, is that, we know that the blueprint of life is encoded in DNA, right? So we know that whatever is encoded in that information polymer determines the functions that the organism will have. Whatever is encoded in a sequence of DNA that encodes for a protein determines what the protein's function are. But the combinatorial possibilities when you're talking about a polymer of size, say 1,000 bases, where there's 4 options at each site, that's basically 4 to the 1,000th power, right? So that's astronomical. And so the question for evolution is, how do you search through that essentially infinite space? And there are very clever things that the evolutionary process does, leveraging both the scale of evolution, where you can have, millions, or I suppose millions is small, billions to trillions of individual organisms searching that space. And so that scale is a big deal in evolutionary approaches. And there's also the fact that there are evolutionary processes, such as how much you drift, the kind of exploitation, exploration trade-offs that you can leverage in order to find, efficiently search through an infinite space. And so I suppose the other end of it is engineering, where typically, or at least the pure form, is that you have like some physical model of how a protein will work, and then you can predict what a change will do to the function, right? But really, we're not very good at that. And I think even though we've become much better at it, the fact that you're looking at an infinite search space is going to mean that you're never going to completely solve this problem. I mean, maybe quantum computing or something where you can actually simulate the exact structures of proteins and the exact functions of proteins will get us there. But it's quite a tall order that deserves multiple approaches. And my favorite is kind of doing what nature already shows works, which is evolution, but at a faster rate and with the control that we have in a lab.
Go ahead.
Oh, so I was going to say, some of the things that I do generally are looking at trying to understand all those genes that we don't know their function. And so there's a lot of diversity, like Chang spoke of, but there's also, a lot of tools that we lack to dissect those functions genetically. And so, expanding our kind of search space in the sense of using microbes that are naturally fix nitrogen or methanogens, or have a specific function we're looking to study, and expanding the number of those microbes we study in order to figure out what all the genes in a given host do. I think is part of kind of being able to do what Chang said in that second part, is be able to predict from some sequence space what those genes are doing, or what the functionality of an organism is, or its capabilities. So.
Yeah, I think I agree completely with both panelists. I think one of the key point I wanted to bring that these are unbiased technologies which act as a hypothesis generating machine sort of, because I don't think so humans are so well equipped to handle the unknown space. So these genetic screens or tools that bioengineers are developing, they act as a hypothesis generating elements, and eventually we go and test them. So doing one gene and one postdoc or one grad student, I think we can have much larger scale, and so that we can forward this science much faster,
Yeah, and I should add that it's not like a competition between the engineering approaches and the evolutionary approaches, right? Like, we all in both camps operate somewhat in the middle, where it just depends on like what area as a lab are you innovating most on. So we're innovating most on the evolutionary side, but of course we start with proteins for which or we try our best to even make predictions on sequences that will get us close to the function without even evolving it, and then let evolution take care of the rest. And I think on the kind of engineering side, there's also that compromise where they're innovating and mostly focusing on like predicting changes to a protein that can make it do something new. But once they have that activity, there's always room for improvement, and then using like evolutionary techniques to make those improvements become a reality. I also want to say that, now in protein engineering and also in genome engineering and systems biology and genomics more broadly, right, there has been this real transformation brought upon by data science, where the idea is that you can leverage extremely massive data sets to kind of predict what might happen. And in some sense, those data sets and the statistical learning approaches on those data sets is basically evolution, where you're cycling through different ways of sampling and combining things and having some way of calibrating or having some way of trying to predict in a high throughput manner what those changes will do, all computationally, right? So I think the most powerful approaches in protein engineering and genome engineering these days are when we're talking about the kind of rational design side of things, is still very knowledge-based. Like Kelly was saying, how they're interested in looking at many existing sources of genes, some of which are unknown, and try to assign function, right? So the fact that nature provides you this large knowledge base means that you can make conclusions from it without having an a priori understanding of everything that is going on. And so that is basically the evolutionary version of engineering on a computer, I would say. So, we're all starting kind of from the middle.
Absolutely. Yeah, so I guess that brings me to a question perhaps for Kelly. So if you're trying to figure out what a gene of unknown function does in a bacterium or another organism, and you can't use other examples to figure out what it does, you can't do some kind of computational prediction, you might do some sort of genetic engineering and manipulate the gene and break it or change it to figure out what it does. So I was wondering if you could touch on maybe what are the ways that we're currently able to control genes or regulators of genes in that way? And what are we still, or what are you struggling with that you might like to do that is kind of, maybe the next type of tool that might be useful?
Yeah, so in the Arkin lab, and also Vivek works on this, and another lab, the Deutschbauer lab, we try to develop high throughput genomic tools that we could apply to many microbes. And we're actually even also trying to apply these to archaea, because if you look at it, any given genome, at least, a third of the genome, we don't know what those genes do. And that's often talking about E. coli, one of the most well-studied microbes. And so when we try to look at, microbes with more diverse function, we, our tools don't always work. Our the way we, make mutants doesn't always work. The way we want to, we try to find, predict what these genes are doing based on prior knowledge, but a lot of the times, computational programs aren't sufficient to do that. And so what we generally do is look for a phenotype. And so can we disrupt genes on some high throughput way and then say, okay, well, this hypothetical protein or this domain of unknown function or this gene that we don't know what it does has a phenotype in a given condition. And from that, can we predict what that gene is doing possibly, or a role it's playing? And sometimes we look at where that gene is located within the genome, what genes are next to it, or if it has phenotypes in many different conditions. And so from there, we begin to try to dissect away and gain knowledge about what those hypothetical genes are doing or those domains of unknown function are doing. And then we can, re-annotate or reapply that knowledge to, microbes that we've already kind of annotated and or, look forward. And Vivek developed some really nice tools to do overexpression of genes in a high throughput capability to begin to look at genes that, from a host, maybe we can't manipulate that host because we can't make mutants in it, but we can put its DNA in another host and overexpress it and see if we can do some sort of gain of function analysis. But I mean, I think a lot of the tools that we have we barely hit on a very small sliver of the diversity that we can go after. And so, I think archaea are a really cool branch of life that, we haven't, we don't know what 70% of the genes do in the most well-studied archaea. And so building tools to be able to rapidly dissect that, because all our annotation tools are based on bacteria or eukaryotes, and archaea are a special, niche within, depending on if you believe the 2-tree or 3-tree branching of life here. But you can't always apply the knowledge you have from bacteria and eukaryotes to those archaea. And so developing tools to be able to dissect that genetically is primarily what I would, am doing and will continue to do. But also, can we build up a big enough database and have enough phenotypes and characterize enough mutants that we can begin to apply that knowledge to microbes that we pull that sequence out of a metagenome? And so those are the kind of questions we look at.
Just to add on what Kelly was saying, I think one of the challenges, if I go into the field and isolate a bacteria and sequence it, about 60 to 70% of the genes are of unknown function, and the remaining ones are all predicted by bioinformatics approaches, which are themselves faulty because they are based on a data which is developed somewhere else or based on some different organism. So we lose the context. So it's a huge problem. There's a gene function gap is a huge problem. It's extremely complicated for a phage or a bacteriophage, again, bringing back around 95% of the genes of a phage completely unknown. So if we are thinking of any application, unless we fill this gap and there is a substantial investment, it's a very difficult to, unless you have money for foundational technologies, you can't really go beyond an application. Just to give a concrete example, if I'm a metabolic engineer trying to produce some chemical, all of the tools we have or screening technologies are mainly color-based or GFP-based. So I think the measurement is one of the things I hope the new era will bring new technologies to measure high throughput in a droplet fashion so that we can do all of these technologies, not only fitness-based, whether the cell survives or dies, or does it show color or no color, but there are so many other applications which need much more elaborate measurements. I think there's a lot of technology necessary, innovation necessary to do that. Right now, it's not there.
And maybe to incorporate a question we got from the chat, how do you think AI and machine learning might be incorporated, especially, if we're still struggling sometimes to be able to do the types of approaches that you're talking about in the wet lab to screen for function? Are we getting better at predicting what proteins do as all the, AI has come on board and data science is getting involved? Has that changed? What do you think the role will be going forward? I guess, Chang, you could answer this, or really anyone.
Sure. Yeah, I'll take a stab. So I guess there are, of course, a lot of forms of AI and machine learning, but perhaps I can reduce it to the type where, at least in the simple case, we have some sort of classifier, or we have some sort of objective function that we can classify performance on. And then we have a bunch of data that is binned into various categories of performance, right? So, take image recognition. That's a pretty well-developed field where you say, this is a person, this is a tree, and then you have a lot of labeled pieces of data, and then you throw more data on it, or it learns from those labeled data, and then it will try to figure out what unlabeled data classifies as. So if we take that analogy to biology and proteins, right, what we know about proteins is that they do something. So, if you see a protein, at the very least, you know that it's, for instance, folded. If you see a protein nature, and perhaps you can even go further and say, this is contributing to the fitness of the cell. And perhaps you can go further and say, this is doing a particular chemical reaction because I've already known that other versions of this protein do that same reaction. So now I'm able to say, I have a bunch of cases where this sequence, this set of sequences does a particular thing, and then I'm going to try to learn the hidden variables from that correlation, right? And so there are two kind of pieces of this puzzle that need to be there for it to fit all together. One is you need to know and have the ability to, in new cases, what a protein does, right? And so you need like high throughput technologies for measuring, as what Vivek said, what the performance of a biological entity is, right? And then on the other hand, you need a lot of data that you can throw at it and make those measurements on. And what is nice in nature is, of course, we have that data from the diversity of life. And so we can leverage that immediately. But what's even nicer, I think, going forward is that because of these innovations in genetic engineering, all of which are very much focused on scale and throughput, right? Such as CRISPR and multiplex editing and barcoding molecular entities, so that you can do, instead of one experiment, you can do 1 million experiments at the same time. All of those advances in genetic engineering are going to allow us to make more perturbations and measure their outcomes in higher throughput. And I think that's where like the statistical learning approaches will thrive. If you can basically tell me that all of these millions of different versions of something do all these different things or perform differently on a particular objective, then I can find out kind of the underlying structure of that correlation and maybe make predictions as to what new changes will do. And on the one hand, maybe you can even understand it in some kind of human way of defining understanding, where it's reduced to some very low number of dimensions, where we know that the principal components responsible for this correlation are A, B, C, and we can design things based on that pretty rational way of thinking. But there's also the possibility that you might just find patterns that are real, but that we just can't hold in our brains, and you actually have to have a computer infrastructure to make those predictions for even new things. And then this is where kind of the AI and human thinking will start merging and blending, hopefully for the better. So I guess that's a very broad answer. I mean, in terms of more specific things, like going back to this antibody engineering case that I mentioned for developing COVID therapeutics, right? So when we evolve new sequences that, for instance, bind and neutralize a target, because our evolutionary systems are so scalable and powerful, we don't just get one sequence, we get tons of different sequences that do the same thing, right? And so there we automatically like meet this criteria of when it becomes effective for statistical learning approaches to be used. And then in future work, very immediately, we're collaborating with machine learning labs to feed that data in and try to make predictions of even superior sequences that kind of take the lessons learned from each individual sequence and try to combine them in some way. And then you can imagine that when machine learning predicts these new sets of sequences, because we have this high throughput capability of synthesizing sequences, we can now remake those and run it through not the computational physics engine, but reality, and see what actually works and actually doesn't. And then that will generate even more sequences based on those predicted computationally that can then feed back into computation. So it's a really nice cycle that you can get into, where you generate data to train models that then predict new sequences and data that you can then use as the basis for generating even more data, all the while improving the functions you want. And maybe in some ideal world, eventually solving the problem of how to design a protein to do anything, even without understanding, you know, in the traditional sense.
Yeah, just wanted to add a couple of points there. That other than a protein, I think at a concrete examples, and we struggle with all of the time in the lab, is can I predict using tools that we have and data that we have, can I predict which changes I need to do to produce a chemical at a higher level? Or can I predict which phage binds to which bacteria? Can I predict how many changes I need to do a particular function? And right now, those predictions are not they are not really good, honestly. And the reason is because we don't have a good data. And the question becomes, what is a good data and how much do I need so that my predictions start falling in place? So for example, I work with Joint Genome Institute, where they are doing a Earth-Virome projects where they're isolating viruses, samples from diverse environments, and they're trying to associate a virus and the host. Of course, there are predictions, there are rules associated to predict their associations, who infects whom. But they are all based on some kind of a predictions and which are not validated. So we don't have really technologies to validate at that level, a diversity level. So the question becomes, maybe the next generation technologies will solve those, right? And in the case of metabolic engineering example I gave you, so right now the question becomes, what should I measure such that my predictions are better? Right now, the measurements are not really good. Can I do metabolomics or transcriptomics? All of the integration of omics, does it give me a predictability? And the answer is still no. And we are still technologies people are developing. We're trying to solve that. But I think there's a huge space for AI and machine learning algorithms, starting with asking a question, how much data do I need? And what kind of data do I need to start, asking the predictability question?
And I would add to what Vivek's saying is, do we have all the metadata associated with your data training set? Like, we have to make sure everything is universal and accurate and large enough in order to build that training set to then apply to new microbes or new. So I think that's where we're, on the genome level, probably not there yet, but definitely I think what Chang was talking to on the protein level, I think there's a lot still to do, or that can be done now, I think, with some of the knowledge we have.
Right. One of the things that I find very exciting about working in bioengineering is that, by far, life is the most complex thing that exists, at least that we know of, right? And so when we typically teach biology or, anything teachable in a course requires you to be able to say like A, then B, then C, then D, right? And you don't want to have too many letters of the alphabet. You want to, be able to reduce it to a number of critical things that explain most of the picture. But it could very well be the case that certain biological behaviors are so complicated that you just, it's not sufficient to reduce it to like a number of variables and links that are easily teachable. And so that's why I think evolutionary approaches, statistical learning approaches, and high throughput biology approaches become more and more important, and they are really going to be necessary in order for us to meet the actual complexity of real life. I may be wrong. It would be nice if biology were actually this low-dimensional thing, and then we're just missing it all this time. And someone will realize that, oh, everything can be explained by 3 or 4 variables. But I don't think that will be the case.
Yeah, and I also want to just point out that I think a lot of what we're talking about is high throughput, data intensive sort of experimentation. And a lot of this is because most people here work on microbial organisms that grow and reproduce really quickly, or individual enzymes that you can evolve in a microbe. I think one of the interesting things that genetic engineering developments in the last few years have enabled is some of these more basic one gene at a time type questions in non-model organisms and cell types that don't grow really fast. So I think just wanted to add, like, I think CRISPR has been used in over 200 different organisms so far, and I haven't tracked talons and zinc fingers, but they've also been used in a lot of diverse cell types and organisms. So I think there's still something to be said for doing a little bit of a slower approach in these organisms that we couldn't study at all before. So we're a little bit behind in understanding how they work, but there's potentially huge value in studying something like an axolotl that can regenerate its limb when it gets cut off, or a blind cave fish, or any of these random sorts of cell types and creatures that you might want to study. So that kind of brings me to a question from the Q&A, which is, I think we're talking very nitty-gritty about different types of technologies, but what is the actual goal of doing all of this genetic engineering? So what kind of changes might we want to see at the societal level that could happen in the next 5 to 10 years as a result of all the types of advances that we're talking about? And obviously there's so many different areas where this can happen, but maybe what are you guys the most excited about?
Yeah, I think I'm excited about a bunch of things, and one of them is microfluidics. I'm hoping microfluidics is operational in the lab as it looks in the paper that people publish, that I want to use it tomorrow, and it should be like on the go, it should be working. A second technology I'm excited about is a single cell transcriptomics, like RNA-seq. Can I measure things in a droplet? Can I measure not only transcripts, but also the chemicals around that cell? In this case, I'm talking about just microbial cells. And then we apply genetic engineering for a variety of applications, and all the drugs that we are producing from microbes, antibiotics, or other tools that we are using in our daily life are from genetic engineering applications. And I think if we want to really go to the next level, we start with the technologies we have. We have this biosecurity thing hanging on our head. We should be very mindful of. So there are a bunch of technologies how to make this safe and useful at the same time. I think I'm excited about all of those areas in general.
Yeah, I mean, I think if you ask all of us individually, right, we have certain projects dedicated to very specific goals, whether it's developing a therapeutic for a disease with resisting resistance in bacteria, or like discovering some new explanation for like how some biological mechanism works. So certainly the goals are like very numerous, and I suppose the kind of broader way to look at it, in my opinion, is that we're really transitioning as I suppose the human race from this era where biology was kind of an observational discipline, right, where you observe what's going on and try to appreciate it. And now it's becoming engineering discipline, where the substrates of biology and the life forms that we use in the lab are actually engineerable, and we have control over them. So it's almost as if like humanity now has this new building material, this new substrate that they can work with, right? Like when, you know, I suppose iron was discovered, or like cement was figured out how to make that, right? So kind of akin to these things, and the application space becomes really broad as a result. I mean, to me, obviously the most kind of privileged applications spaces when you're working with biology as a substrate are going to be applications that influence biology. And so therapeutics is definitely top of my list on this, right? I mean, the goal of therapeutics is to cure life of disease, people, plants, animals, etc. So biology as a substrate already has a privileged role in that. So if we can start engineering biology better, then therapeutics is a big societal win from that angle. And another thing that has to do with life is, of course, like ecosystems, and how do we maintain organismal diversity, and how do we resist climate change? Things like this have, you know, or biological substrate has a privileged role in. And so those two areas are kind of where my favorite applications lie. Not because, you know, you can't come up with some really neat like electronics chip that uses biological parts and perhaps has new properties, but it seems that the privilege of having this new substrate is that it already knows how to interface with life. And so those are going to be the problem areas that we can get the most bang for our buck from.
Yeah, I mean, I think I would also say some of the plant microbe interaction stuff, you know, trying to get away from having to make a ton of fertilizer. You know, 2% of our fossil fuels goes to fertilizer. And so if we can, you know, create a better, you know, microbiome for the plant root system, you know, we can reduce the amount of fertilizer we need. I mean, there's also, you know, a project working on, one of my projects I'm funded to work on is to, you know, support manufacturing for deep space exploration, and kind of basically use bacteria to make the things you would need to go to Mars, including like, you know, fertilizer for plants, or make acetate, you know, to grow other microbes for bioremediation or decontamination of regolith, or, you know, just to recycle any components that we use and put them back into a closed system. And, you know, one of the cool things is if we, you know, get really good at predicting, you know, what, you know, drugs we need or what proteins we need to synthesize, you know, in theory, we could have, you know, DNA synthesized on Mars, and that transformed into a bacteria or a yeast strain. And then that yeast can make the medicine that that astronaut might need on demand. And these are kind of big, crazy ideas, but these are things we need to kind of think about what we understand from the engineering side now for planning for missions, you know, 10, 20 years away now for like deep space research. So I think that's some of the cool, like, you know, applications that are less, you know, immediate, but, you know, need to be thinking about now for the long term.
Yeah, and I just want to emphasize on what Kelly's saying. You know, I love this idea of like using biology in space. The reason it's a good idea is because biology is self-organizing and self-replicating, right? So it requires very little infrastructure, as we typically think of, to boot up. You know, the instructions are self-encoded, they're autonomously manifesting, and they can autonomously replicate if you provide it very simple ingredients. And so certainly for like space missions, this becomes a very good idea because you are limited in what you can bring to space, but also terrestrially, it's also will have as great impact because there are resource limited environments all over the world. If you give poorer locations the ability to make their own drugs on demand, that becomes a big social change. And if you just make it easier to access these, let's say, therapeutics or more complicated functional materials that are biological, if you make it easier to do that, you kind of democratize the infrastructure and the ecosystem of production, right? So that you don't need to rely on, you know, $100 million lab facility to be able to generate therapeutics. You can do it cheaply, and then many labs can do it all simultaneously. And if that barrier of entry is low and more people do it, you have a faster pace of innovation as well. So I think it's kind of a special discipline when we discuss engineering with biological matter rather than, you know, your kind of traditional materials that you think about.
Yeah, that's a good point. And I'll add the things that I'm the most excited about for the next 5 years or so. And what's been happening right now is really human therapeutics that have been developed as products of genetic engineering. So CAR T cells, as an example, have been really transformative in cancer therapy. So this is letting us engineer T cells or white blood cells to be able to attack tumors and find them and destroy them. And this has been kind of taking off in the last couple of years, and I think will continue progressing in the next 5 years. And then the other therapeutic is gene therapy products. So we've seen a couple of gene therapy products approved by the FDA very recently, and I think this will continue. And gene therapy is a little bit of an older technology than some of what we've been talking about, but it has kind of only recently been materializing into a good outcome. So it had some dicey early days, but now we're able to use viruses that are not harmful to package and deliver useful genes to people therapeutically. So if you're missing a gene that helps with your retinal function, for example, there are people that can see now because they've been delivered this gene. So to me, that's really exciting. A lot of the CRISPR-based therapies are just being tested now, and early clinical trials are looking very good. So sickle cell disease is kind of the shining example so far of how CRISPR has been used medically. And we have one or two examples of individual people that basically are cured for the time being, at least, of their symptoms of sickle cell, which is really incredible, but something we have to watch in the long term. And so I think based on, or piggybacking on what something Chang alluded to, this democratization concept, I think in general, that's definitely true, that a lot of these genetic therapies or genetic tools let people all across the world do really exciting biology in their labs. I get really concerned when I think about these things that I'm most excited about, the human therapeutics, and think about how much they cost. So in the US, we haven't exactly solved healthcare. We don't need to get into that, but so far, when we've seen approved gene therapies, they're millions of dollars or $500,000, or these really just incredible price tags. And it makes sense in some ways, because this is in theory a one-time treatment that will prevent all the future medical costs that someone might have. And it can be very expensive to manufacture the virus that delivers the reagents. But that's concerning to me. And I've read a lot of cases of people that can't get their insurance to cover it. And so this is not helping them at all. They can't access it. So I was curious what everyone thought about the economics of what we're doing and the eventual products. Is it just very different in the therapeutics world versus kind of the more industrial microbial engineering world? Is it our responsibility as scientists to be thinking about this upstream? What do you think about that?
I can take a stab at it. I think I've been thinking about it a lot, especially when we see these products like human gut microbiome. Okay, you can get a kit and you will get a diagnosis of what is happening. And as a scientist, I feel worried about seeing some things which are not really honest or trustworthy, because we are not there yet understanding exactly what these microbes are doing, who's good, who's bad in that microbial pool. We don't even understand what are their functions, and really whether the disease modalities that we see is because of a particular microbe. So once we don't know that, I think selling these products to people at a price tag, whatever it is, it's going to be extremely is a negative impact on the science itself, because it's not going to solve the problems that you're facing. I think I'm not sure, I'm not expert in the CAR T cell and other technologies, but I think at least microbial diagnosis, I think, or the production of a chemical, I think those are all industries still trying to understand what is the price tag and how much it is valuable or economics of that. But I think if you are using a microbe for a probiotic, for putting it in a soil or giving it a human, I think we can still need to get a level up in terms of economics is a secondary. I think science itself needs to be really validating those technologies. If I take a probiotic, is it really good, or does it do what it is saying it is doing? Then the price tag will come later on. I think we are still at that level where there are some products which are really crazily priced, which are not really doing the job. So I think as a scientist, I'm really worried about those issues.
I mean, Megan, I really like the point you made about gene therapy, which is that, you know, if and when done right, and certainly all my bets are that, you know, it will just completely revolutionize therapy in the next 10 to 30 years, let's say. But when done right, it should be a permanent correction to a particular disease. And that means that you pay for it once and you don't have to pay for it again, right? Whereas many drugs you have to take all the time. And that will be economically very important. I mean, I suppose you know, my view is that if things can be cheaper, of course that's going to be good. The kind of you know, maybe counterpoint to it is that, at least on the local time scale, right? If you're promising something that's very cheap, then you know, there's not as much profit to be made. And so that one could argue, reduce the level of innovation, right? People talk about this for antibiotics all the time, where, you know, antibiotics compared to like a protein therapeutic that you have to take once a month for the rest of your life is peanuts in terms of the amount of revenue you could potentially generate. But they're potentially just as important, if not more, given what Vivek was saying earlier about how, you know, that'll be the predominant mode of death, or at least exceed cancer if we don't deal with it now. But I think those concerns are not so bothersome to me, because as long as you know, if the result is important enough, right? You can not only have pharma working on it, you'll have, of course, academia working on it, but you'll have like philanthropic agents put money into it, and you'll have government agencies invest properly in these developments. And then once they become very cheap, you may still have some period under patenting where you can derive a pretty large profit on them. And then it's correct to have it then just be passed on and be free to society after that. And then you'll basically be forced to make the next innovation, right? So like this idea of driving things to be cheaper and cheaper with these new biotechnologies is, I think, always a good thing. You know, it's perhaps there are going to be some like local challenges in terms of getting the motivations to be at the right level, but ultimately it'll be a major accelerator of innovation.
Yeah, I just wanted to add also that, you know, when I look at the insulin prices, which is a product of genetic engineering that we see, is crazy in the US. Where I go to Canada or in India, for example, where I'm from, the price range is totally different. So the people who are suffering here, they should be getting benefited as the world is, everybody is getting. I think there's a whole set of a whole different separate section for a panel. We need discussing what are the, you know, pros and cons of technology per se, but also I think government needs to be, you know, more involved in these kind of issues, particularly pricing for antibiotic or discovery for antibiotics. Right now, the pipeline is drying up, not only just for the economics, but also there is no incentive for the companies to start inventing new drugs. and whether they sell it better. I think there are two different things. One is the insulin kind of an example, or an antibiotic discovery. I think, and CAR-T is another spectrum of that price range. I think we need more investment and also policy changes at the level where, patients are benefited at the end of it because they are paying the taxes. The research is funding the, is inversely proportional to what is happening right now.
Right, and
Absolutely.
because
Go
of
ahead.
this lower and lower barrier of entry to biological research, and concomitant democratization of it, you don't need to rely on large companies that have profit as a major driver of their decisions to make new drugs, right? And we can already see that today, where large pharma companies have shrunk their discovery platforms in service of basically buying smaller companies to fill that role. And so the reason I think that there are all these smaller companies that can come up is because there's a shift in kind of the infrastructure necessary to do biological research. It's really become a lower barrier of entry.
Yeah, absolutely. I wanted to turn to a question in the chat that I think is interesting. So Timothy asks, in terms of innovation, how much biological complexity is needed to make meaningful biological conclusions? So we have a variety of model organisms that are popular, but predictability and reproducibility in biological processes remains low. So how much of our modern biological advancements are limited by the models that we've been using? And maybe Kelly, you sort of touched on this in talking about microbes that we can't culture or we can't easily manipulate in the lab. Do you have thoughts on how much is out there that we're really missing?
I mean, I think we're missing 99% of it. And I'm talking about just the microbial space. But I think modern biological advancements are themselves tools. We're building new tools, and that's an advancement. And then can we adapt those or apply those to new microbes that are non-model microbes? And I think, at least for microbiology and bacteria and archaea, it's really been, as soon as we can isolate it, and someone is interested in a specific function, can we dissect those functions and build tools, and whether we're characterizing them with microbial physiology and validating their function based on sequence prediction, or we're trying to better understand all the genes in that process. And so, that's a lot of what I do, and some of what Vivek does in our larger group is design and build tools to dissect and to move away from just, our E. coli lab strains, but really going out and grabbing, the methanogens that are, making 80% of the atmospheric methane, and then, trying to tame them in the lab. Or, cellulitic biofuels, was kind of a big thing a while ago, and trying to find microbes that naturally, eat cellulose, and just trying to find that diversity out there. And, new genes and new processes that we haven't yet discovered or don't even understand. I think we're trying to bring those into the lab and dissect those traits as best we can. And then that goes to Vivek's phage, him bringing in, as many phage he can to characterize, and move away from lambda phage and just the specific phage that we classically study.
Yeah, it's totally. I think we have such a lack of, we have been focusing on the economics, and which is not let's say biofuel, an example. Everybody was trying to do E. coli or yeast as a tool for producing a biofuel. Maybe it is not a good idea. Maybe there are other bugs which produce better intermediates, which we need a minimal engineering. And that is lacking just because we don't have genetic tools developed. Or a gut microbiome, I want to understand gut microbiome. What are the model organisms? None of the tools that we have work in the gut microbiome because the tools we did not invest in those tools. I think the CRISPR-I is definitely one of the best tools we have, but I think even using that at a power that is potential that it is still lacking. We need more in terms of investment and also just looking at the problem space.
And do you mind explaining what CRISPR-I is briefly for those who might not know?
Oh yeah, so CRISPR-I is a you know better, Megan. I think I can just attempt it. I think CRISPR-I is essentially you're not using the CRISPR-Cas system not to cut the DNA, but to have the Cas system goes and just station there at the locus where the gene is suppressed, expression is stopped. So you can assess gene function by just down regulating the expression of a gene and asking a simple question, okay, what happens if I regulate the expression of a gene using CRISPR?
Right, yeah, and in a lot of microbes, if you cut the genome, they're just going to die. So you can't really do genome editing in the same way you might with a human cell type. So yeah, using CRISPR-I, you can activate a gene and tune up expression or tune down expression. There's also CRISPR-A, which is the activator, which will tune up expression.
Yeah,
so
I think this is
a whole suite of tools.
Exactly, and this is especially important for addressing essential genes. You can't really delete a gene which is essential for organisms. So can we tone down a little bit and see how that impacts the physiology of the cell?
I think there's a lot of discovery there we still need, because there's, CRISPR doesn't work in many, many microbes as, rapidly as other tools do. So, building up our CRISPR-I toolbox, and again, building up our toolbox to dissect these non-model organisms is key.
Yep.
So I want to make sure we get the questions answered. There was a kind of old one for Chang. Asking, when you have so many mutations, when you're sampling this huge sequence space in one of your experiments, how do you know when you have the right product? So maybe you could describe selection.
Oh yes, of course, right. So the the most powerful version of a directed evolution experiment, or especially one that is based on our hypermutation systems, is one where you can couple the property you want out with the fitness of the cell, right? And then it becomes a totally autonomous process where sequences that, beat some bar will get an advantage in their replicative fitness, and then they will increase their numbers, and then their descendants, which make changes in the sequence of their parents, will more likely improve even further, right? So this happens over and over again in an autonomous fashion, and that's evolution. But you bring up a good point, which is that there are not, what you want a gene to do, a protein to do, may not be what the organism cares about. Certainly there are a lot of exceptions where it is exactly what the organism cares about, and you can make a very easy selection. But when it's not as obviously linked to what an organism cares about, then you can design kind of like a genetic circuit that synthetically links the function you want to what the organism cares about. And that capability of designing good genetic circuits and robust ones that can link an arbitrary function to cellular fitness, that capability has expanded greatly because of synthetic biology. And so, with some cleverness, you can usually think of something and get it to work. And then going back to what Vivek was saying about microfluidics and other high throughput characterization methods, those you can use a physical device to look for mutants that meet some criteria that you can then enrich. And the key is that, because we're exploring through mutation and selection or trial and error, you just have to have everything work at a massive throughput. And so these physical devices that are based on, sorting and microfluidics have also come a long way to becoming higher and higher throughput. So you can solve this problem in a number of ways.
Great. And we have another question that is a little bit beyond my depth. I think it's for you, Chang. Do you see a link between evolutionary biology and reinforcement learning in working together to create more promising search policies?
Okay, it may be also a little bit beyond my depth too. I think, well, you know, the history of this is, I mean, I suppose if you look at genetic algorithms, right, they are based on the rules of evolution. They are based on kind of the evolutionary trade-offs between exploitation and exploration. They're based on the idea that there are often optimal mutation rates. They're based on the idea that, you know, the strength of selection you impose at any given point may influence the later nature of the outcome. They're also based on the idea that you sometimes want to limit your search space so that you can stochastically sample things that aren't optimal at some rate because there are greedy search algorithms that don't get you to global optima, right? So all of these ideas influence genetic algorithms. And I think there's some link, of course, between genetic algorithms and current reinforcement learning. And there's also some link between genetic algorithms and, you know, what we're trying to do in my lab, which is kind of to make it so that the evolutionary systems that genetic algorithms were inspired by can now be revitalized in the laboratory, but with the same level of control that you can do in a computer in terms of the parameters. So I do think there's a link, and I think the kind of search policies that are developed in one side do apply to the other. I mean, I think that like many search policies or kind of the heuristics for them are like reasonably well established. And so it's a matter of implementation. And certainly in a computer, you can control parameters much more finely than you can do experimentally. So for us, it's very important to figure out how to experimentally, through engineered systems, have more resolution on how to control the parameters that go into an evolutionary algorithm. Yeah, I mean, but I think I'm missing a lot in terms of this link as well, because, you know, I focus on genetic algorithms, and there are all sorts of different statistical learning architectures that are relevant today. So I'll leave it open.
All right. I think we have 3 minutes left. So if you had one pressing final question to add to the chat, go for it. But otherwise, I guess I will bring it to the panelists. Do you have any closing thoughts that you'd like to share about any of the topics that we've discussed today, or what you're excited about, where you want to see more investment, anything like that?
I just wanted to add that, you know, I think his generation has been like asking who's there in a microbiome or entire space that we are in, whether it is inside us or outside us. In a microbiome space. So I think we need more understanding to where do we go from here, other than asking who's there, but what are they doing? How are they doing? So that we can think about applications, not only phage, but also microbe interactions and microphage interactions, and complexity with the human context itself. Like how do they interact with the microbe-human interactions?
So yeah, I would say, you know, along the next line, you know, sequencing is really cheap and easy. It's really easy to do metagenomics and just throw a ton of money at sequencing genomes. But, you know, I think developing tools to dissect what, you know, the genes that we don't know what they're doing, especially when we move away from the model organisms, is really key to having a better understanding and being able to engineer these microbes to do the functions we want later, or be better at making molecules or performing a specific task.
And Chang, maybe in addition to that question that just got asked, is what is the #1 challenge with the work that you do?
Okay, so I suppose I can interpret it two ways. One is like, what's the thing that we're always struggling with? And then the other is, you know, what's kind of our priority in terms of a goal we want to achieve. So I'll try to answer both. You know, the main challenge, I suppose, is that I guess this is a bit broad, but forgive me. I mean, it's just always surprising to me how little you can predict what a change will do in like some biological system you're working with, right? So, you know, of course, our engineered genetic systems, we have pretty good understanding of how they work, but everything is experimental. Like even the most obvious conclusions or obvious inferences you make in the biological world can often surprise you, right? So I think we're just very limited in terms of how much predictive capabilities we have. So that's a main challenge. I suppose other challenges come up day to day is just like, you know, getting your DNA on time, like getting the company that makes it to deliver it earlier and right, like make the cycle quicker. In terms of like a big goal of ours, certainly right now, we're pretty heavily focused, although we haven't published on it yet, but we will soon, on antibody engineering, because that's just a very, it's like a tried and true therapeutic molecule. And I think we have ways of directing it to bind and neutralize numerous new targets that at a scale that people didn't have before. So that's a big challenge for us. Yeah, and I'll leave it at that.